Okay, let's think about this for a moment. Your brain, right? It runs on neurons firing, well, kind of slowly. We're talking maybe 10, 20 milliseconds for signals to pass between them.
Yeah, relatively speaking, that's not exactly lightning speed, is it?
Not at all. But then, how do we manage to, react super quickly, dodge something coming at us, make those split-second decisions that are literally life or death sometimes?
That's the big question, isn't it? It's this really fascinating paradox. How does this, well, biological hardware, which seems a bit sluggish component-wise, pull off these incredibly rapid and complex computations?
Exactly. It feels like it shouldn't work as fast as it does.
And a key part of this is the new framework they introduced called CHARM. That's CHARM.
Right. Complex harmonics decomposition. Sounds complex.
It does, but the insights it offers are pretty compelling, suggesting how the brain might achieve this fast, sort of spread out computation despite those slower neurons.
So our mission today, basically, is to figure out how the brain's structure, its wiring, and its activity patterns all come together to allow for this speedy, non-local processing.
Exactly. We'll be looking at how CHARM, especially when applied to this huge neuroimaging data set your sources
Mm.
mentioned, helps reveal what's going on under the hood. Hopefully, we can uncover some real aha moments.
Okay, let's dive in. This puzzle. Slow parts, fast system. How does the brain square that circle?
Well, the thinking is shifting away from just neuron-to-neuron chats, the really local stuff. The key seems to be in distributed computation.
Distributed computation, so like the whole brain working together.
Pretty much. Think of it less like a chain reaction and more like, I don't know, an orchestra playing together. It's the coordination across large areas that matters.
Okay, and this shows up in things like resting state networks. I've heard that term.
Right. You get these large-scale patterns of coordinated activity. What's interesting is that these patterns, these networks, are actually much simpler, lower dimensional than the raw brain activity you'd see with fMRI or EEG.
So it's like the brain is simplifying its own complex activity into these core networks. How does that happen?
The idea is that it involves something called critical dynamics. It's a bit like the brain being poised right at the edge of order and chaos.
Poised,
Yeah.
like ready to tip either way quickly?
Exactly. That state is thought to allow signals or influences to travel much further and faster across the network,
Yeah.
enabling those long-range interactions needed for these simpler whole brain networks to emerge.
So it's not just signals hopping to the next neuron. It's about this balanced state allowing for broader communication.
Precisely.
Yeah.
And the brain's physical structure is perfectly suited for this.
Hey.
Your sources mentioned the BAW plan, the basic architectural plan of the mammalian brain.
Okay.
It's mostly short-range connections, regions talking intensely to their neighbors. But crucially, there are also these sparse but incredibly important long-range connections, expressways, basically.
Ah, okay. So like highways connecting distant cities while most traffic stays local.
That's a great analogy. And it's this mix that makes the brain's network quite different from, say, a simple crystal lattice or other physical systems. Understanding how these non-local effects, driven by both the critical dynamics and these long-range physical wires, create the networks we see is, well, fundamental to understanding cognition.
Right. Which brings us back to charm. You said it helps capture this non-locality. How?
Okay, so CHARM complex harmonics decomposition. It's designed for what they call low-dimensional manifold reduction. Fancy term, but basically,
Simplifying the complex data.
yeah, boiling down that super high-dimensional brain activity data into its essential underlying network patterns, finding the core structure in the noise, you could say.
So finding the main themes in the brain's activity.
Kind of, but how it does it is different. Many traditional methods use math related to the heat equation. Think about how heat spreads gradually to nearby areas.
Okay, so good for local stuff.
Very good for local relationships, yeah. But CHARM, it's based on the Schrödinger wave equation.
Wait, Schrödinger? Like quantum mechanics, Schrödinger? What's that doing here?
Uh-huh. Yeah, it sounds unexpected. But the mathematics of wave equations, like Schrödinger's, are really good at describing things that interact over distances and create interference patterns, like waves in water.
Okay. I think I see waves travel and interact.
Exactly. So the mathematical tool CHARM uses, derived from that equation, is inherently sensitive to these long-range, wave-like interactions in brain activity. Especially when you factor in those critical dynamics and the long-range physical connections we talked about, it's built to see those non-local effects.
That's actually really neat. So it's looking for patterns that ripple across the brain, not just spread locally.
Precisely. Your sources have a figure showing this, taking the complex data, breaking it down into these simpler manifold networks, and another figure compares it directly. You see PCA looking at variance, other methods using that heat math, seeing local stuff, and then CHARM capturing both local and non-local.
And they tested this with models too, right, to see if it really picked up on the non-locality.
Yes. They used whole brain modeling specifically to check how well each method captured effects known to be non-local, like the influence of criticality in those long-range connections.
Okay, makes sense. So how did CHARM stack up when they threw actual human brain data at it from those 1,000+ people?
Right, a massive data set. And the results were pretty interesting. First, they looked at static functional connectivity, basically, a snapshot of which brain areas tend to fire together on average.
Like a basic correlation map.
Sort of, yeah.
Yeah.
And on that measure, CHARM, PCA, and the other harmonic methods, they were all pretty similar. They could all reconstruct that static picture reasonably well.
Okay, so far so level.
But then they looked at something more dynamic. They used a measure called edge-centric metastability, ECM.
Edge-centric metastability, what's that capturing? The wiggliness of the connections.
Wiggliness is a good way to put it. It measures how much the strength of connections between brain regions fluctuates and changes over time. It's about the dynamics, the variability.
Gotcha. And how did CHARM do there?
That's where it really shone. CHARM significantly outperformed the others in capturing these dynamic changes, this ECM.
Whoa, okay. So for the static picture, they're similar, but for the changing dynamic picture, CHARM wins.
By a clear margin, according to the study. And this strongly suggests that those non-local effects, the long-range interactions and how they evolve, are really, really important for actual brain function. It's not just a static map.
That makes intuitive sense. The brain isn't static, right? It's constantly changing. So CHARM's better at seeing that dynamic dance. But why? What's the mechanism?
To dig into that, they used whole brain modeling again. They basically built a computer simulation of the brain based on average connectivity data.
From the 1,000 participants.
Yep. And they tuned the model, adjusting parameters like the overall connection strengths, until the model's activity best matched the real fMRI data.
Okay, standard modeling approach. What did they find?
Something really cool. The model best matched the real data when its global coupling parameter was at a specific value. And crucially, this same value was where the model showed the most Kuramoto metastability.
Kuramoto metastability. You said earlier that's like a proxy for criticality.
Exactly. A good indicator that the model brain is in that balanced critical state. And at that specific critical point, CHARM was way better than the other methods at correlating the model's underlying activity dynamics, its ECM, with the dynamics in the simplified network patterns it extracted.
Wow. Okay, let me try and rephrase. When the model brain was acting most like a real critical brain, CHARM was best at linking the raw activity to the important network patterns.
You got it. It's strong evidence that CHARM excels precisely because it captures the non-local effects that are prominent during criticality.
And what about those long-range physical connections? The highways.
They tested that too. They made two versions of the model. One with the normal, sparse, long-range connections included, and one where they artificially snipped them out.
Ah, a virtual lesion study, kind of.
Yeah, sort of. And then they compared how well CHARM versus the other methods captured the difference those connections made to the brain's dynamics, again, using that ECM measure.
Let me guess, CHARM was better.
Significantly better, yeah. It was much more sensitive to the amplification of dynamics that those long-range connections provide. The difference in ECM correlation between the with and without models was much larger when analyzed with CHARM. It really highlights CHARM's ability to pick up on the impact of the brain's specific wiring.
Okay, this is building a strong case. CHARM seems uniquely suited to seeing how criticality and the brain's structure enable these complex, large-scale dynamics.
It does seem that way. And they took it another step further by looking at different brain states.
Like awake versus asleep.
Exactly. Wakefulness versus deep sleep. They applied CHARM to brain activity data from people in both states.
And presumably the brain activity looks pretty different.
You'd expect so, right? And CHARM definitely picked that up. It revealed really distinct patterns of interaction between its simplified networks during wakefulness compared to deep sleep. The functional connections between these core networks were significantly different.
Your sources showed visuals of this, like different flow patterns between networks, more active when awake.
Yes, precisely. The flow of information or influence between these networks looked quite different. So different, in fact, that they could train a machine learning model just on the CHARM network interactions, and it could tell with high accuracy whether the person was awake or asleep.
That's pretty impressive discrimination.
It is. And other measures changed too. That dynamic variability, the ECM, measured within the CHARM network space.
Mm.
It's significantly different between wake and sleep. Same for the directedness of the interactions, how much information seemed to flow hierarchically.
So it's not just capturing dynamics in general, it's sensitive enough to show how those dynamics fundamentally change between mater states like being awake or deeply asleep.
Right, and one more interesting wrinkle here. They found that the link between the raw brain activities dynamics, source ECM, and the dynamics in CHARM's simplified network space, manifold ECM, was significantly stronger during wakefulness.
Stronger when awake. What does that imply?
Well, the suggestion is that the awake brain operates closer to that critical state we discussed earlier. It exhibits more critical dynamics than the brain in deep sleep, and CHARM, being sensitive to criticality, picks up on this stronger relationship during wakefulness. Other methods didn't show this difference as clearly.
That makes a lot of sense, actually. You need your brain to be more responsive, more dynamic, more critical when you're awake and interacting with the world.
So wrapping this all together, the big picture from your sources is that CHARM, with its mathematical roots in Schrödinger's equation, seems like a really potent tool. It helps us see the non-local, dynamic, and often critical nature of brain activity much better than previous methods.
And it strongly implies that these underlying network patterns that finds these manifolds are not just some mathematical curiosity. They're fundamental to how the brain actually computes things so quickly with its slow parts.
That really seems to be the main takeaway. It's a shift away from thinking computation happens just at the neuron level or even
Right.
just within distinct brain regions. It pushes towards seeing computation as an emergent property of these large-scale interacting networks.
So the networks are the computational engines, especially when the brain is in that critical dynamic state.
That's what the evidence seems to point towards. These manifold networks appear to be a core organizing principle for whole brain function, driving the critical dynamics that let us think and react so effectively.
It really is an aha moment at that paradox we started with, slow neurons, fast brain food. Maybe the answer lies in this incredibly sophisticated network coordination, these non-local effects that CHARM helps us visualize. It's the system's dynamics, not just the part's speed.
Exactly. And it leaves us with a really provocative thought, doesn't it? If we can get better at understanding these manifold networks, how they work, how they change.
Yeah. Could that unlock new ways to understand brain disorders? Maybe conditions where these network dynamics go wrong?
Potentially. Or, looking ahead, could it even give us clues for building better AI? AI that computes more like a brain, leveraging these kinds of distributed, dynamic, non-local principles.
Imagine that. AI based not just on connections, but on the dynamics of those connections, operating near criticality. That's definitely something to chew on.
It really makes you appreciate the complexity and, frankly, the elegance of how the brain tackles computation. This deep dive feels like just scratching the surface of criticality, network science, and what neuroimaging can reveal.
Absolutely. A fascinating journey into how our own brains pull off their amazing feats. Thanks for breaking down CHARM and all these
It's
ideas.
a pleasure. It's truly remarkable stuff.